Welcome to Marn's Biology. Today's topic is recombinant human insulin. First of all, what is human insulin? The human insulin is a peptide hormone produced by beta cells of pancreatic islets, encoded by insulin gene. It regulates the metabolism of carbohydrates, fats, and proteins by promoting the absorption of glucose from blood into liver, fats, and skeletal muscle cells. In the tissues where absorbed the glucose that is converted into the glucogen. The glucogen is a storage material and it utilizes for a further energy process. Human insulin is present in chromosome 11 and the human insulin protein is composed of 51 amino acid chain, A chain and B chain. The A chain composed of 21 amino acids and B chain consists of 30 amino acids. The both chains are joined together by disulfide bonds between two cysteine residuals. And the insulin was discovered by Sir Edward Zarpy Zucker in 1916. And the human insulin is produced by a recombinant method is called cumulin. How the recombinant insulins are constructed? First of all, that insulin hormones have two chains, alpha and beta chains. The alpha chains and beta chains of beta cells were isolated and that inserted into the BBRT22 vectors with beta glucosidase. The beta glucosidase acts as a marker. First, we take the BBRT22 vectors, then cut it with restriction enzymes. Then the proinsulins, the two chains are called as a proinsulins. The proinsulin chains are inserted into the vector. Then the chains are ligated with the DNA ligase. Then a plasmid was inserted into the E. coli for application by fermentation method. In purification process, that fermented E. coli cells was taken and the cell lysis and recover the inclusion bodies and that lysed pellet and washed the inclusion bodies and its solubilization. It undergoes several chemical process to extract the proinsulin. The two chains are extracted, the alpha and beta chains were extracted. Then it go to the sulfonamide method and became enzymatic cleavage and it two chains are separated each other and it became constructed and the two chains are constructed to form a active insulin. Recombinant host produced that proinsulin chains. The fused beta galactose A chain and B chain separately, and that proinsulin A and B are separated from a beta galactosidase from a treatment of cyanogen bromide. The detached proinsulin chains where a beta galactoside is possible because of extra codon forms a methionine was added at the end terminal of each gene for A and B chain. After detachment, A and B chains are joined in vitro to form a native insulin by sulfonyl peptide chains with sodium disulfonate and sodium sulfate. Diagram shows that the two chains of alpha and beta chains are inserted separately into the BBR322 vectors with beta glucosidase gene. The diagram shows that alpha and beta chains are inserted separately into the two vectors with beta galactosidase genes. The each chains are replicated via E. coli genome, and that it purify the genes, and the two chains are joined together by sulfide bonds and become active insulins. Applications. The insulin causes cells in liver, skeletal muscles and fat tissues that take up the glucose from blood. In liver and skeletal muscles, the glucose is stored as a glycogen, and in adipose tissue, it is stored as triglycerides. Insulin stops the use and inhibit the release of glucagon hormones. The persons with the diabetes 1 depend upon insulin shots. The most commonly insulin is injected in the subcutaneous from patients that's a hormone that no longer produced in bodies. The diabetes 1 is also known as the insulin dependent diabetic mellitus. Thank you and subscribe that Modern Biology for further videos.